So on the left is a polypeptide. You can see the backbone, it's a string, and it looks blue at one end and red on the other. That's just a carboxyl end and the amino-terminus. But the little branch structures coming off of it are the side groups of the amino acids, so we can see those. We don't know what each one of them are. I mean, we don't even care what they are. But this polypeptide, it's a little bit misleading, because I don't think that it's true that a polypeptide gets made and released by a ribosome before it starts folding. I find that very hard to believe. Nobody knows the answer to that, BTW, but it's probably not true. Okay? But in this model, it is true. And so what we're going to do is watch both sides of the screen at the same time. And this will be our polypeptide struggling, searching through different folding conformations to see which ones it can find that are lower energy. And then this is the energy landscape over here, where the trough in blue represents this lowest energy state, and red would be the highest energy state. And there's other pockets that are in this folding landscape that represent lower energy. But this is, for example, a lower energy than the red, but it's not the lowest energy that the protein can adopt. And then the energy that's currently possessed by the system, which includes this polypeptide and the water molecules surrounding it, is represented by the hockey puck. So as this polypeptide searches different shapes that it can adopt, the puck will move around on the surface of that energy landscape. If it adopts a lower energy state, that should coincide with a drop of the puck and presumably some sort of structure in the protein. So we'll watch it once or maybe twice if we feel that it's helpful. So watch the folding of the polypeptide in the puck. So it's pretty high energy state now, but now it's adopted a slightly lower energy state. It's actually in this trough or halfway around it. And what you can see is that the protein's starting to adopt some secondary structure that's helped to reduce the energy. And that includes an alpha helix here and the formation of a beta sheet up there. That polypeptide is now in a lower energy state than it was as a linear polypeptide, but clearly it's got a lot of amino acids that are still exposed to water, and this one, tryptophan, we know is a hydrophobic amino acid, so it's not happy in water. And there's another big ring structure and a long carbon chain coming off a couple of others. So I don't think it's quite in its final form yet. We can watch that, continue to watch it. It's now heading down into a lower trough, so I'm suspecting we're going to see a lot of folding happening quickly. And you can see that the structure is becoming globular, which means some of these big side chains look like they're moving to the inside of the protein, escaping water. And so we know that that's going to reduce, through this hydrophobic effect, it's going to reduce the energy of the system. And so that looks like what's probably happening there. And we can continue to watch this thing. And now it's down in the bottom trough. And now you can see it's a globular protein. There's still some pretty good sized side groups sticking out into water, and I'm sure they're not all hydrophilic. That might be one of those, that might be glutamate, for example, where this end would be charged, but I know that those would be saturated carbons, so they can't like the water. But you're never going to find a protein where all of the hydrophobic or non-polar side groups are stuck in the middle of the protein. It's just impossible to do that. So some of them can be sticking out. Likewise, there might be a polar group that's buried inside of the protein too, but predominantly they'll be on the outside. And now you can see that there's actually 4 beta sheets involved in that final structure, and there's an alpha helix that goes across them. And now you're in the lowest energy state. The protein can't get more stable than it is there. Okay. So I don't know that there's a point in playing that again. We watched it. for that, I am done with protein structure and stabilizing forces. And we're going to now move on to function of proteins. How do they acquire function? And what you'll find is that the same principles that we've been discussing apply to function as well. So I'll remind you what a polypeptide looks like. You can model it any way you want, with the side groups being stick figures or balls for the backbone or space filling model. Space filling model to me is the most satisfying one. It's the more realistic description of a protein, because it allows you to understand that there aren't a lot of spaces inside the protein, but there are a lot of surfaces on the outside. And you're not seeing alpha helices here. We know they're formed in this polypeptide. What you're seeing are the electron density, an electron cloud around that polypeptide. And so this is what I describe as the nooks and crannies. The reason I like that is because it allows you to think about possible interactions of this protein with anything else, whether it's another protein or whether it's a substrate. It's got to fit the protein. Right? It's got to form interactions with that protein somehow. And the nooks and crannies are an easy way to visualize that, right? Or a hand in a glove kind of thing. The two molecules have to be complementary in some way for them to form stable interactions. And we know what those stable interactions are probably going to be. They're not going to be covalent forces, but they're going to be all the other things that we've thought about. All those other four forces. So let's take a look at some of those ideas. I know I described the cytoplasm of a cell to be much like a cesspool at one point. The whole portalette thing that you guys didn't really take to, but that's actually true. And it's a very crowded environment. And molecules are moving around. It's not like they're just laying there floating on an ocean. It's not like a bottle floating out in the ocean. Molecules are moving around. They're much, much smaller than bottles. They have less density, they have less mass, and they're easily moved around by forces that are known as Brownian motion forces. And that's really just the heat of a system causing vibrations and movement between molecules. So if you were to jump in a glass of water, if it was ice cold water, if it was ice, then the molecules of water aren't moving, right? They're frozen in space and time. But as that ice thaws, those water molecules now that are free of the ice begin to move around. They might be moving really slowly at refrigerator temperatures, but at room temperature they're moving quite a bit, actually. And certainly when they start boiling, you know that that's utter chaos inside that liquid water. So our bodies are about 37 degrees centigrade, 96, 97, 98 degrees Fahrenheit, pretty warm. Molecules are moving around. And as a result, they're bumping into other molecules. If we believe this idea that a protein has mostly nonpolar groups on its outside, on its inside, and polar groups on its outside, it's actually being shielded by water a lot, but it'll be bumping into other molecules. So then the question is, what happens when it bumps into another molecule? What happens when one protein bumps into another one? The motion of the molecules is going to make sure that it's bumping into a lot of molecules all the time. They either bounce off one another or they stick to one another. Those are the only choices that I'm aware of. The only way they can stick to one another is if the forces holding them together are greater than the forces trying to separate them. And the forces trying to separate them are the other molecules bumping into them. Okay, so we know that two molecules won't stick to one another through a single hydrogen bond because we all appreciate that those are weak. We know that they're not going to stick together by a single van der Waals interaction because we know that's also weak. Ionic interactions in an aqueous environment, we know those are pretty weak. So what are we left with? This hydrophobic effect, maybe? So most molecules, when they bounce into one another, are going to bounce apart. And that's what this figure is meant to represent. We've got a protein molecule A bouncing around inside the cell, and it actually encounters three other molecules called B, C, and D. A is never going to form a covalent bond with any of them spontaneously by itself. If they happen to be both substrates that are grabbed on by an enzyme, so there's a third molecule that grabs them both and brings them in and then does a chemical reaction, then you can form a covalent bond. But two proteins bumping into one another, or a protein and a substrate bumping into one another, is never going to be joined by a covalent bond unless there's some enzymatic activity associated with it. We're not talking about that yet. So then the only forces that could hold them together are these non-covalent forces that we've been talking about over and over and over again. And the point of this slide is to say, look, a lot of those interactions are going to have chances of forming permanent associations, but most of them are not going to be stable. And so A with B, well, we can count the non-covalent interactions there. There are three. We don't know what they are, but it doesn't really matter. They're non-covalent, and therefore they're weak. And in this cartoon, they're not strong enough to hold A together with B. And so that would be an example of two molecules bumped together, maybe sit there for a fraction of a second, but then release and move on, or easily disrupted by a third molecule bumping into them. Likewise, A with C, that's just one more non-covalent interaction. That's not going to form a stable complex either. But now if you look at A and D, now those, let's see, that would be a nook and that would be a cranny, I guess. And that's a nook and that's a cranny. And see, every nook and cranny seems to fit together really nicely. The shapes of those molecules are such that they can form lots of interaction sites. Now, just because you fit together doesn't mean you're going to stably associate with one another. But if in those nooks and crannies there's interactions, then those could form stable associations. There's hydrogen bonds in there, there's ionic interactions that are possible in there, there's certainly van der Waals forces that are in there. If those are all non-polar side groups, their desire to get away from water, this hydrophobic effect, could also drive the association. And so A will find a partner, likely, in the cell eventually. All proteins do something in the cell, so there is a partner for every protein. That partner might be a substrate, might be glucose, might be lactose, but there is a partner. Okay? And so those interactions will occur. In this case, A is naturally meant to find D. It might take some time, but when it finds D, it buckles onto it. But it's going to bump into lots of D's and C's along its route, and it's not meant to interact with those. There's not enough covalent interactions to make that a stable complex. And so those are examples of every other protein in the cell that it's just going to bounce off of. Okay. We're going to look at some very specific examples. So we start with this cartoon, but then we've got to get into something a little bit realistic, and we will, as soon as I find my pen. Okay, we're still at the level of a cartoon, but now we're looking at an enzyme, and this enzyme is shown in green. So this is a protein. In this case, it does something. It binds to a substrate. Now, for us, hey, we could put names on these if we wanted to. We could say this is beta-galactosidase, and that would make this lactose. That's an example that we've at least put some time thinking about. And this is a real interaction that we know occurs in bacteria. So how does it occur? Well, there must be a binding site, these nooks and crannies that are present on the folded structure of beta-gal, which I hope you understand has lots of alpha helices and beta sheets, doesn't just look like a green blob. But it does have a binding site that perfectly matches the ligand such that those two molecules can interact and form a stable complex. An enzyme that's going to take lactose and convert it into glucose and galactose has to hold on to lactose. There's got to be a stable formation of a complex of some sort between those two before it can act on it and create products. And so while we're thinking of that and have that picture in our mind, you should be thinking that, well, what happens when it does break that bond? Now you don't have lactose anymore, you have glucose and galactose. And the answer is, you're right. Beta-galactosidase doesn't like holding on to glucose and galactose. The non-covalent interactions are weakened once that glycosidic bond is broken. And that's how the products come off the enzyme. It can no longer hold on to them as tightly as it can with lactose, so it releases them. Glucose and galactose float off, leaving that binding site now ready again for another lactose molecule. You grab another lactose, cleave it, can no longer hold on to the products and releases it. Binds another lactose, so it's like an open and closed active site. And beta-galactosidase can convert hundreds of lactoses to glucose and galactose per minute, hundreds of them. So it's a very fast process. Even though, as we'll see in a minute, it's pretty complicated. Okay. Oops, I don't know what I just did there. Okay, so we've been talking about protein structure a long time, and they don't just fold. Once they're folded, they do something, and it's the process of folding that allows them to do something. That folding positions their amino acids in certain places in space that they now can do something. So we're going to look at a very specific example. We're building up to something. So here's a polypeptide, and I'll put the amino terminus down here and the carboxy terminus up here. This is a long polypeptide. I don't know how many amino acids are in there, but I'm going to say 100. We're not looking at 100 of them, but I believe there's 100. Here's one. It's a serine, an arginine, a glycine, a glutamate, a serine, a threonine. They're not side by side in the primary structure. You can see the serine's way down by the carboxy terminus, and the threonine and the serine down here are way down by the carboxy terminus. And there's tens, 20, 30, 40 amino acids between them. But that protein folds after coming out of the ribosome into a globular structure, and what you can see is that this protein that's folded now has an active site or a binding site for a substrate that's formed by the folding of the polypeptide. And if you look at these select amino acids after folding, they look very different than they did before folding. After folding, this serine here was a long way away from this serine, but after folding, now they're side by side in the final structure. The arginine, which was 20 or 40 amino acids away from the serine, is now practically touching the serine. Okay. So this should come as no surprise to us. We're aware that the folded structure could bring amino termini next to carboxy termini in the final structure. We've looked at examples of that, so that shouldn't surprise us. But now we've got an active site, and so now we're going to think about what happens in this active site and how does it happen. This protein is going to be our model protein for most of the rest of the semester that I'm here, and we're going to call it protein kinase A. That's the name of this protein, pKa, also known as pKa. I'll put that in parentheses. We're going to think about this protein a lot because it'll be our model for not only protein function but also signaling pathways that we're going to talk about towards the end of the semester. So protein kinase A, kinases are molecules that do one thing. They take the terminal phosphate from ATP, oops, not ATR, Dan, ATP, and by the way, this is ribosomal ATP as opposed to the ATP that's used to make DNA. It's the energy carrier in the cell. It takes the last phosphate off of ATP and puts it onto another protein, phosphorylating it. and as a byproduct forms ADP. So this is adenosine triphosphate, adenosine diphosphate. The difference between those two is one phosphate group, which this kinase, and all kinases do this, has now taken from ATP and put on to another protein, some random, it's not going to be random, but some protein that has yet to be named. That's what kinases do. That's a whole family of proteins that all they do is take a phosphate from ATP and put it onto a protein. Why so many kinases? Because we've already talked about this idea that most proteins only do one thing. So the one thing that a kinase does is recognize the substrate and put a phosphate on it. The next kinase also puts a phosphate, but it's a different substrate. Right? So the difference between kinases is the substrate that's recognized, not the fact that they move a phosphate onto it. All kinases do that. The question is, what's the substrate? So protein kinase A has actually a couple of substrates, but there's 1,000 kinases in your body, all recognizing different substrates, different proteins that they will move a phosphate onto. We'll talk about what the effect of a phosphate group can have on the activity of a protein, but not today. Okay, I'm going to get through this next slide for sure. So this is still protein kinase A. I'll draw the protein as we saw it in the last picture. Okay, so and I'll quickly zoom back. We're talking about this protein here. And now we're looking at the functional groups of these amino acids in the active site. So now we're looking with a high-powered microscope that doesn't exist. So I've drawn the back, it's kind of the globular structure of the kinase itself, but now we're looking at the serines, the threonines, the glutamic acids that were only shown as balls in the last figure. And now we're looking at their ability to bind a molecule known as cyclic AMP. Cyclic AMP is a really cool molecule in the cell. It's called a second messenger. Second messenger. Messengers we'll talk about more when we talk about cells communicating with one another. Messenger, second messenger. So these concentrations fluctuate in the cell depending on what's happening outside the cell. Anyway, here's a molecule of cyclic AMP indicated by the brown structure. If we look at the active site or the binding site for cyclic AMP in protein kinase A, we saw on the last slide that there was a serine. Well, here it is with its hydroxyl group. And notice that this is a hydrogen that's covalently attached to an electronegative atom with another electronegative atom nearby. And so that's our definition of something that can form a hydrogen bond. And so this is our hydrogen bond, forming between the serine residue and one of the oxygens on the phosphate in cyclic AMP. That one interaction is not strong enough to hold cyclic AMP in place. But here's another one. Here's arginine with its positive charge interacting with the negative charge on an oxygen attached to that same phosphorus. Here's an amine group, clearly on the backbone of the polypeptide. That's a hydrogen attached to an electronegative element. Nearby another electronegative element. That makes that another hydrogen bond. Here's glutamic acid. That's a charged amino acid, but it's not using its charge in this case. It's using its electronegative oxygen to hydrogen bond with a hydrogen on an electronegative atom. So that's another hydrogen bond. Here's threonine. Threonine, we haven't looked at the structure, but it looks a lot like serine, except it's got one additional methyl group. So we know this hydroxyl group is a polar bond with oxygen having a partial negative charge. It can form a hydrogen bond with this amine. And this amine can form a hydrogen bond with the serine. And the hydrogen on the serine can form a electrostatic interaction with this nitrogen. So now we're looking at 1, 2, 3, 4, 5, 6, 7 non-covalent interactions that together allow protein kinase A to bind to cyclic AMP in a stable way. Sorry. I'm in trouble talking, thinking, and writing at the same time. So how do molecules bind to other molecules? Well, it's exactly the same stupid thing we've been talking about for 3 weeks now. So I hope that at least you're seeing the same thing over and over again. It's like almost becoming boring, but at the same time it should become interesting because it's used so often by so many molecules and of such great importance. I know it's 1:31. Friday, the exam is at the beginning of class, I should think, right? And it lasts for whatever time I said it lasts for. I forget. I'll have to look back at my rules. Whatever that was, that's what we're going to do. And then we'll continue with our final lecture until we all go home for some extended period of time. And I'll have some news on the laboratory by Friday. Okay? So thank you. It's nice to see you.